And thank you to everybody for getting up at this ungodly hour to come hear about something you might learn something about. All right. Before we get to the science, though, I just want to mention a couple things about the society. And that is that the society spends a fair amount of money on what we call discretionary activities. And these range from education and outreach to awards and grants to sponsoring workshops and symposia on things like professional development. We have a public policy committee which gets in touch with legislators. We have a diversity committee to try and increase the diversity along many axes of members of the society. So these are activities, and many, if not most of these, are actually run by society members. So if you're interested in getting involved in any of these activities, you have an opportunity, or will have an opportunity when you renew your registration, to check a box to say, I'm interested in maybe education and outreach, and I'm willing to volunteer some time to help with that. Okay, so please take advantage of that if you can. Now, a second point I want to make about this is that I think it's important for society members to understand the financial aspects of the society. So to that end, in a couple of months, we're going to begin posting yearly reports on society budgets, income, and expenditures. Okay? And so look for that on the society website in the next couple of months. And then finally, I want to mention, this is about half our budget. The other half goes to sort of fixed costs. And so we have somewhere on the order of a $600,000, $650,000 budget each year. And a lot of that goes out in terms of awards and grants. And just dispersing that money is an enormous task, okay? And currently, our executive vice president, Andrea Case, is doing that job in addition to all of the other jobs that she does and the things that she's responsible for. Okay? This has become really a tremendous burden on her or whoever is going to be in that position. And so the council has recommended that we establish a new position in the society. And that's a position of treasurer who'll be responsible for the financial matters of the society. Now, the council just can't do that by fiat. That's going to require a constitutional amendment. Okay, so you will be asked to vote on a constitutional amendment creating the position of treasurer with the next round of elections that we have. Okay, so let's turn to the science. What I'm going to talk about today is a project that we've had going on in our lab for a few years now. And this has truly been a collaboration between myself and 4 other people so far. 2 former graduate students, a current postdoc, and a current graduate student. And Joanna, Kate, and Irene have or are all giving talks on matters related to this area, so I hope you have or will go see what they have to say. Okay, so just a little refresher, okay? Allopatric divergence occurs when we have one species or population whose range gets split into 2 or more pieces by some sort of barrier that prevents migration between those populations. These populations diverge from each other due to selection and or genetic drift, and then the barrier breaks down, allowing the ranges to overlap in what we term secondary contact. And if reproductive isolation is not complete at that point, then of course gene flow can occur. So historically, evolutionary biologists have been interested in a number of questions about gene flow in zones of secondary contact. Questions like, how are species boundaries maintained in the face of gene flow? How does gene flow affect reproductive isolation? One can get either reinforcement or fusion, theoretically. How does gene flow affect morphological divergence? How does gene flow influence genome structure and organization? And can gene flow in sympatry actually facilitate allopatric speciation? What I'm going to tell you about today are some studies we've done to address some of those questions. I'm going to talk about three things. One is how gene flow homogenizes allele frequencies in sympatry. I'm going to talk about how gene flow affects character divergence. And I'm going to talk about how gene flow affects reproductive isolation. Okay, so let's start with homogenization of allele frequencies. So just a little bit of reminder. So for any loci with divergent allele frequencies, gene flow between populations is going to tend to homogenize the allele frequencies, right? So with neutral alleles, gene flow, if it's extensive enough or lasts a long enough time, is going to reduce or even eliminate allele frequency differences. For divergently selected alleles, okay, in that case, if we start with population differences in allele frequencies, gene flow will tend to cause those frequencies to converge, but it might not do so completely because selection is resisting that. Okay. So those are sort of the expectations. And because of that, what we can get if we look along the genome and we look at degree of divergence between two populations or species, we might find many areas where there's neutral variation or neutral differences among the species. And if gene flow goes on long enough, we'll reach a drift migration equilibrium where there's very little difference. On the other hand, with moderately divergent selection and migration, we can maintain some level of divergence between the populations in the face of gene flow. And if there's very strong divergent selection, then we can see very large differences despite gene flow. And this slide shows you why I didn't become an artist. Okay. So now this kind of differential divergence across the genome has been reported numerous times, a lot of recent studies. But there's much less evidence, there's not much evidence that this difference in divergence across the genome is actually driven by variation in the strength of divergent selection. And so that's the kind of issue that we were interested in looking at in our lab. Okay, so let me tell you a little bit about the study system. We're looking at two species of morning glories. These are indeed sister species. Ipomoea cordata triloba is a mixed mater. It has larger, more pigmented, showy flowers with more nectar. Ipomoea lacanosa is highly selfing, and it's evolved many of those characteristics that are typically associated with what's known as the selfing syndrome. Smaller flowers, loss of pigmentation, loss of nectar production, loss of scent, and so on. Okay. The selfer, lacanosa, is found mostly throughout North America. That's the pink county records here. Cordata triloba, the more outcrossing species, has a more southerly distribution. It even extends into Mexico, but we don't have county records there. And then there's sort of a region right in here where the ranges of the two species overlap. And at some sites within that region of overlap, there are actually both species occurring together in sympatry. And they're occurring, essentially growing intertwined with each other, right? Because these are vines and they climb all over everything. Okay. Now there's also partial reproductive isolation between the species in the form of cross-incompatibility, which is prezygotic or very early post-zygotic. So you can see if you cross either the two parental species, you get 4 seeds per capsule. That's the maximum. If you cross the two species with each other, there's a substantial reduction in the number of seeds produced. There's also an unknown amount of post-zygotic isolation because we haven't even really looked at this very much yet, although we do know, we do have a BDM incompatibility that we've mapped to 2 different chromosomes, and this incompatibility results in infertile pollen. And it appears to be, from what we've done so far, fairly geographically widespread, but more work needs to be done on that. But otherwise, we know very little about, excuse me, about how much post-zygotic incompatibility there is. Okay, so for our study, this is the genetic data we collected. We did leaf transcriptomes of each species, 31 different accessions of one and 30 of the other, and this generated a total of about 67,000 SNPs. 27,000 of them were synonymous and 21,000 were non-synonymous. The rest were in non-coding regions. And if we look at population structure, K equals 3 is the optimal number of groups here. And what we can see is, regardless of whether lacanosa, the sulfur, is in sympatry or in allopatry, it's basically all, the same collection of genotypes. Just one. On the other hand, if we look at chordata triloba, what we see is that the sympatric individuals tend to be one genetic type, and the allopatric individuals tend to be a different genetic type. So 2 distinctly different populations. And this one site of allopatry indicated by these 3 arrows, there's other evidence to indicate that this was probably recently sympatric as well. Okay, so let's look at some evidence for gene flow between these 2 species in sympatry. And we can use either measure pi, this is actually pi times 1,000, or average gene frequency difference between the loci. And what we can see in either case is that there's much more divergence between the species in allopatry than there is in sympatry. On the other hand, if we look within each species, sympatric and allopatric chordata triloba are quite different from each other, whereas sympatric and allopatric lacanosa are very similar. Okay, same pattern no matter what measure you use. And this is all consistent with the structure pattern that we saw in the previous diagram. And what this points to then is gene flow being unidirectional, more or less, from the sulfur, lacanosa, to the outcrosser. Okay? That's what we suggest accounts for this big difference, this big difference here between sympatric and allopatric chordata triloba. Okay. So what kinds of SNPs actually introgress? This is I think, an amazing diagram. Let me walk you through it, okay? So on the x-axis, what we have here is for a particular SNP, okay, we have the allele frequency difference in allopatry. So we calculate allele frequencies in allopatric populations and in sympatric populations, okay? On this axis, okay, we have the allele frequency difference between the species where they're in sympatry. Okay? And so each SNP gets categorized into one of these cells, okay? And then the y-axis is just the percent of those SNPs that are in a particular box. And the way this is portrayed is that these row frequencies sum to 1, okay? And if there were no gene flow, what we would expect to see are populations lying along this line right here, which is that the difference in sympatry is equal to the difference in allopatric frequency, okay? And that's not what we see at all. What we see is that for SNPs that have frequency difference between species below about 0.9, we get effectively homogenization of those SNP frequencies in sympatry, okay? Whereas for allele SNPs that have allele frequencies in allopatry that are 0.9 or above, there seems to be some sort of resistance to homogenization. And one might expect that that resistance is due to divergent selection. Okay, so a couple of inferences so far. One is that gene flow seems to be of sufficient magnitude and has gone on for a sufficient amount of time to homogenize what we assume are mostly neutral loci, okay, that have lower frequency differences. And as I said, this suggests that the highly diverged SNPs are actually resistant to homogenization because of divergent selection. Okay, another way to look at that is just to look at admixture. In the sympatric chordata triloba, where admixture is just the proportion of alleles derived from each of the two species. And if we look at the less divergent SNPs, those with a divergence of less than 0.9 in allopatry, what we see is, I should say 69 instead of 66, is that in fact, in chordata triloba, the more outcrossing species, actually at any given locus, on average, 69% of the alleles are derived from the other species, okay? Whereas only 17% of the highly divergent SNPs have the alleles derived from the other species, okay? And if we look, however, at sympatric lacanosa, okay, we see something completely different. Basically, all of the alleles there are lacanosa alleles. There's essentially no introgression from, no admixture from chordata triloba. Okay. So, we can actually ask then whether these highly divergent SNPs, which seem to be resistant to homogenization, are actually subject to divergent selection, and we can do that with a standard MK test. And we can see that whether we take specifically completely diverged loci or loci that have diverged by a frequency of about 0.9 or greater, in both cases, it's highly significant. And we can estimate the proportion of these loci at which alleles were fixed by selection to be somewhere around 50% or so. So pretty good evidence that this resistance, apparent resistance to selection, is indeed due to divergent selection acting in sympatry. Okay, so let me summarize what I've said so far. There seems to be significant, mostly unidirectional gene flow in sympatry, and that's consistent with what people have found in other systems comparing an outcrosser with a selfer. Gene flow tends to go from the selfer to the outcrosser, basically because if you're a selfer, selfing preempts ovules and prevents outcross pollen from doing anything effective. There's significant allele frequency homogenization for less divergent SNPs. There's high admixture in chordata triloba, the more outcrossing species, for less divergent SNPs. On the other hand, there's a resistance to homogenization by highly diverged SNPs. Little admixture in sympatric chordata triloba for those highly diverged SNPs. And highly diverged SNPs do appear to be subject to selection, at least a large proportion of them. Okay, and so that's at least consistent with this picture that I showed before of moderate and strongly divergent selection causing regions of high divergence between the genomes. Okay. So now I want to move on to the next topic, which is character convergence or divergence. So let me give you a little cartoon explanation of what the expectations might be here. So if divergence in characters is neutral, that is to say the alleles underlying the character divergence are neutral, then what we would expect is that the amount of gene flow that we saw in the previous part of the talk should cause these characters to converge, characters in chordata triloba, to converge in sympatry to those that we see in allopatry. I'm sorry, those we see in the other species, lacanosa. Whereas if character divergence is due to divergent selection, then there won't be complete character convergence because selection is opposing that. If we have very strong divergent selection, then we won't see much convergence at all, and we'll see a strong species main effect in an ANOVA. On the other hand, if there's weaker divergent selection, then gene flow might pull the character down towards that of Lacanosa, but not get there completely, and we should see a species by location effect. Okay, so the traits we looked at for other reasons were primarily floral and inflorescence traits, okay? 3 of them showed this kind of pattern where there's essentially no convergence in sympatry, very strong main effect. And 4 of them, okay, showed this species by location interaction. And let me show you then what those look like. So in each case, you know, and corolla length, nectar sugar concentration, cyme length, and nectar volume, what we see is a significant change going from allopatry to sympatry in Chordata triloba, and it's in the direction of the phenotype for Lacanosa. And for all 4, we fail to see a significant change going from allopatric Lacanosa to sympatric Lacanosa. And again, this is consistent with the basically unidirectional gene flow that we saw earlier. Okay? So what this again suggests is that floral characters, or many of them anyway, are resistant to convergence in sympatry. Okay? And presumably some of the SNPs on which we found divergent selection to occur in the previous part of the talk, some of those SNPs are underlying these characters. Okay. And we presume at this point that this resistance to phenotypic convergence is due to divergent selection on the phenotypes in sympatry. But if that's the case, that sort of poses a bit of a conundrum, right? Because as I said earlier, in sympatry, these 2 species grow intertwined with each other. It's hard to imagine that, at least with respect to the physical environment, they're experiencing different selection pressures. So one possible resolution of that conundrum is that, in fact, these suites of characters represent different adaptive peaks on the adaptive landscape. And if that's so, then gene flow would tend to move the population down off the peak, but then natural selection would tend to restore it up to the peak. Okay? Now, that's a hypothesis. It's one that can be empirically tested. And if I live long enough, I hope to see somebody in our lab actually test that hypothesis. Okay. Finally, I want to talk about the effects of gene flow on enhancement or reduction in reproductive isolation. Okay, so what are possible effects of gene flow on reproductive isolation? Well, one is that it, as we said before, it can homogenize trait differences causing prezygotic isolation if those traits are nearly neutral, right? And so one kind of example of this might be reduced flower color difference that is responsible for pollinator isolation. Another possibility is that gene flow from the selfer to the outcrosser can introduce into the outcrosser alleles that increase selfing, which in turn would actually increase prezygotic isolation because selfing is an isolating mechanism. Okay? Gene flow could increase prezygotic isolation by reinforcement, or it could decrease postzygotic isolation by selection against incompatibilities, something we might term fusion. So what we want to know is, are any of these happening in Ipomoea? Okay, so first thing I want to look at, I want to return to this cross incompatibility I mentioned earlier. Okay, cross incompatibility acts either prezygotically, so the pollen lands on the stigma, it germinates, it sends a pollen tube down through the style here, okay? And there can be all kinds of interactions between the pollen and the styler tissue that either facilitate or inhibit pollen tube growth. So that could be one contributor to this kind of cross-incompatibility, okay? Or there could be something going on when the pollen tube reaches the ovule, either preventing entry into the ovule or causing death of the embryo, okay? And I just want to point out that if it's occurring at that stage, even though it's postzygotic, this developing seed is still under maternal care, right? The mother is provisioning that seed. And so it is possible, even though it's postzygotic, for reinforcement to occur. Okay. So here's a design of an experiment that we did to ask whether cross-incompatibility differs between sympatric and allopatric, between sympatric and allopatric populations. So we sampled a number of different sites. Some of them were sites where Lacanosa was growing by itself. Others were in allopatric populations of the other species. And then in this small area right here, we also had some sympatric sites as well. Okay, so the crossing design was portrayed here. We have 4 different combinations of the 2 species, right? And whether they were from an allopatric or from a sympatric population. And we took each of these individuals and we mated them to other individuals of the 4 different categories in both directions. Okay. And so what do we see? If we look at mean seed number, okay, and we compare individuals that were collected from allopatry versus individuals collected from sympatry, we see that there are about 10 times as many seeds produced by the sympatric individuals than the allopatric individuals. And that means that there's been a decrease in the amount of cross-incompatibility. Now that could be due to a change in either Chordata triloba or a change in Lacanosa or both. Right? So if we look at just Chordata triloba and we ask what's the average seed set for allopatric individuals versus sympatric individuals, what we see here is about an 8-fold increase in seed production or a decrease in crossing compatibility in sympatric populations of Chordata triloba. By contrast, if we look at Lacanosa, the selfing species, there's no difference whatsoever. Okay. So this decrease in cross-incompatibility seems to be largely, in sympatry, seems to be largely due to a change in the outcrossing species into which gene flow has occurred. Okay? So just to summarize that, cross-incompatibility in sympatric Chordata triloba is reduced, but it's not reduced in Lacanosa. And this, again, is consistent with one-way gene flow, and presumably this has occurred because there's been the replacement of Chordata triloba alleles at loci causing cross-incompatibility with Lacanosa alleles in the sympatric populations of Chordata triloba. There don't appear to be replacement in the other direction, okay? And we certainly don't see any evidence of reinforcement of this incompatibility. Now, that could be due to the fact that postzygotic isolation is weak, but we just don't know, right? Because we don't have much information on postzygotic isolation. So that's another area that we want to follow up on. Okay. So another thing we looked at was the effects of gene flow on selfing rate itself. Right? So selfing, as I mentioned, is a form of prezygotic isolation. It reduces fertilization by heterospecific pollen, primarily, at least in these kinds of species, where the anthers are clustered around the stigma. Okay? Basically, there's preemptive pollinization of the ovules by self-pollination, and any heterospecific pollen that arrives just gets there too late. Okay. In Ipomoea, in general, selfing, the degree of selfing is inversely proportional to anther stigma separation, and botanists have a term for this, like they do for all sorts of things. That's called hercogamy. Okay, and so what we were interested in is how do hercogamy and selfing rates in sympatric and allopatric Chordata triloba compare? And first, what we see is reduced hercogamy, reduced separation of anthers and stigma in sympatric Chordata triloba versus allopatric Chordata triloba. And no differences here. They're already about as close as they can get. Okay. And if we look at selfing rates, what we see is that that decrease in hercogamy is accompanied by an increase in the selfing rate in the sympatric Chordata triloba, up to very near what the selfing rate is in Lacanosa. Okay. So unidirectional gene flow seems to have increased reproductive isolation by increasing the selfing rate in the sympatric Chordata triloba. Now, of course, there are a number of possible causes for this. One is that we might have simple replacement of alleles affecting selfing rate by gene flow. Okay. Another is that actually there may have been reinforcement of this process, adding to the effect of simple gene flow. And the third is that selfing alleles, as is well substantiated theoretically, have a transmission advantage over outcrossing alleles. And so that transmission selection may have also played a part in causing this increase in selfing rates. We don't know the relative contribution of any of these. Certainly they could all be occurring simultaneously. And again, that's an area where we're interested in, pursuing. Okay. And we also haven't proven, okay, but I think it's very likely based on studies in both this system and other systems, that this increase in selfing rate probably does substantially decrease gene flow from Lacanosa to Chordata triloba. And again, that's open to empirical investigation. Okay, so final topic I want to turn to is how these evolutionary dynamics in sympatry might affect reproductive isolation between sympatric and allopatric populations of Chordata triloba. Okay? So how does reproductive isolation arise in very general terms? Well, genetic divergence typically is thought to contribute to the accumulation of reproductive isolation. And we've certainly seen that there has been genetic divergence between sympatric and allopatric populations of Chordata triloba. So could this genetic divergence have contributed to some sort of reproductive isolation developing between sympatric and allopatric populations? Okay, so if sympatric populations came into contact, would we actually see reduced gene flow between these two populations? Okay. So we think, we're pretty confident that gene flow from the allopatric to the sympatric genotypes would probably be substantially reduced because of the high or increased rate of selfing that we've already seen in the sympatric Chordata triloba. Again, we're inferring this, okay? It's not proven yet, but I think it's very likely to be happening. So that leaves the question of what's going on in the other direction. Okay, what about gene flow in this direction? And in particular, we can ask, has any cross-incompatibility developed in matings between sympatric and allopatric populations of Chordata triloba? And here's sort of a schematic way this might work. Okay? So we know there's reduced sympatry between, reduced cross-incompatibility in sympatry between the two species. Okay? And that's because we think sympatric Chordata triloba contains lacanosa alleles that affect cross-incompatibility. Okay? So here we have the two species in allopatry, and we can imagine in a sort of cartoon fashion that there are two loci that cause this incompatibility to be expressed. And if you're little a in lacanosa and little b in Chordata triloba, then little a and little b are incompatible, and so you get an incompatible reaction. Okay. In sympatry, what's happening, we think, is that the alleles from lacanosa have moved or introgressed into Chordata triloba. Okay, and now we don't have any little b's, right? So that's what reduces the incompatibility between the two species in sympatry. But now, Chordata triloba in sympatry is this genotype, and so when we compare allopatric Chordata, or mate allopatric Chordata triloba with sympatric Chordata triloba, we once again have the possibility of an incompatibility being expressed. So the question is, does that happen? And in fact, it seems to. Okay? So here is the average seeds per capsule if we cross two allopatric populations of Chordata triloba. And here's what happens if we cross an allopatric versus a sympatric population, okay, in the two different directions. And what we can see is that there's about a 30% reduction in seed set when we cross with the sympatric rather than the allopatric population. Okay, and so what these two things together suggest is that gene flow from allopatric to sympatric genotypes is probably substantially reduced because of the high degree of selfing in the sympatric Chordata triloba. And gene flow in the other direction is reduced substantially, maybe 30%. Okay? So there have arisen quite a couple of fairly strong barriers to gene flow between these populations. And so, you know, do we have here incipient speciation? Well, maybe. Okay, let me summarize this then. So gene flow has had opposite effects on different reproductive isolation components. This is for interspecific isolation, okay? There's decreased isolation in sympatry that has been caused by decreasing the cross-incompatibility. But there's increased isolation between the species in sympatry because of this increase in the selfing rate in Chordata triloba. So in the long term, what do we expect the net effect of these two opposing components to be? Do we expect fusion or speciation or limbo? And my argument would be that this increase in selfing basically trumps the loss in cross-incompatibility, and what we have are nearly two separate species in sympatry. So gene flow, I'm arguing, and we still need more data to say this convincingly, but I'm arguing that gene flow in sympatry has actually led to almost complete speciation. If we look at intraspecific isolation, okay, the increase in cross-incompatibility between sympatric and allopatric Chordata triloba, as well as the increase in selfing rate in the sympatric populations, has led to the development of substantial isolation between sympatric and allopatric populations. And that's a consequence of the dynamics that's occurring in sympatry. Okay, so let me just tell you a little bit about some future directions we're hoping to head in. So as I said at the beginning, the evidence from the McDonald-Kreitman analysis indicates that about half of the highly divergent loci, or SNPs, seem to be resistant to introgression. That immediately raises, however, a couple of questions, right? Which ones? Which ones are in that half, right? And of course, why? You know, what makes them resistant? And presumably, this has to do with the phenotypes that the SNPs are associated with and selection acting on those phenotypes, right? And so I think what that means is to get any further in understanding this pattern of resistance to introgression, we unfortunately have to start looking at individual loci, okay? Loci associated with the individual SNPs that seem to be resistant. And that is going to be, you know, well, I'm not going to do it in my lifetime, right? But we can at least make a start in that direction. Okay, and so I want to tell you about one project that we're initiating in the lab to address this issue. And it focuses on flower color, one of my favorite traits. And so I want to first compare allopatric chordata triloba with allopatric lacunosa. Okay. You can see that the flower color is very different, and it differs in two areas, in the limb here, as well as in the throat. And it turns out that there are two loci that control that pigmentation. There's a throat locus called T, and a limb locus called L. Now, in sympatry, what do we see? In chordata triloba, we see there's still limb coloration, but there's no throat coloration. Okay? And it looks like the data is pretty good right now. I'm not going to show it to you, that the little t allele has, in fact, introgressed into sympatric populations. Occasionally, in chordata triloba, in sympatry, we actually get individuals that have flowers that look like this. Okay? Flowers in which the little l allele also appears. Okay? But they remain pretty much at quite low frequency. So we have this contrasting pattern where at the T locus, gene flow seems to have homogenized the genetic composition in sympatry, but that hasn't occurred at the L locus, right? And so that, again, suggests that there's resistance to homogenization, or resistance to introgression at the L locus. I can talk about an L locus. There's this whopping SNP that we found for limb color. There's a Lodge score of over 60, if you can believe that, okay? So we're dealing here probably with a single major locus. And if we look in the region of the chromosome, the 1.5 LOD confidence interval, we now have a genome for these species. And we've done resequencing of 20 individuals of each species. And so we can actually look at regions of admixture in the region where this QTL lies. And you know, there are 3 areas where there's very low admixture compared to the rest of that region. So what we're seeing here is an average of about 70% introgression from lacunosa. That's consistent with what we saw before. But there are a couple of small regions where admixture is significantly lower, right? And so those are probably the regions to look at for finding the actual locus that underlies this QTL. Now, interestingly, the T locus is in the same region. We know what that is. That's MIB transcription factor, okay? And again, that T locus, right, the throat locus, seems to be in a region where there's been a high amount of admixture. Okay, so the idea eventually is then to actually perform some field experiments directed at the question of, is the fitness of chordata triloba individuals with colored limbs higher than the fitness of individuals with white limbs, okay? And in particular, we'd be interested in looking at whether there's differential pollinator visitation to these two genotypes. And what we're sort of, our working hypothesis here is that during the period when all this homogenization is occurring, okay, and the reproductive isolation is developing, there's gene flow that introduces the white limbs. And during this period, if bees are visiting those genotypes with the white limbs, but then, you know, moving because there's constancy to the other species, Blacanosa, that pollen's effectively wasted, okay? And so we would expect during that period selection to prevent the introgression of the limb allele from Blacanosa. So that's sort of our working hypothesis. And hopefully in the next couple of years, we'll have some experimental evidence as to whether that's true or not. Okay, so let me then summarize what I've said today. First of all, is that gene flow upon secondary contact has pervasive effects on genetic composition, phenotypic divergence, and reproductive incompatibility. Variation in genetic divergence across the genome appears to be due, at least in large part, to variation in the strength of divergent selection acting on loci in those regions. Morphological differences, at least in the floral characteristics that we saw, morphological differences between species seem to be maintained by divergent selection in the face of gene flow. Gene flow leads to increases or decreases in different components of reproductive isolation, but probably nearly complete reproductive isolation in sympatry in the form of increased selfing. And maybe what I think is perhaps the most interesting finding that we have is that gene flow in sympatry may be facilitating speciation between sympatric and allopatric populations of the same species. I don't have a final slide, but I do want to acknowledge support from NSF and from Duke University. Thank you very much. And I guess there's no bell in here, apparently, no chime, but we have 1 minute. So if there's a question, we'll take it. All right, go to your next, oh, Linda. Yeah, that's my argument, is that that's probably what it's moving in that direction right now. Yeah. So that's certainly an intriguing possibility, right? Is that gene flow in sympatry leads to 3 species rather than just 2. Thank you.